So, bacteria are what are known as prokaryotes. So that means they are single-celled organisms lacking a membrane-bound nucleus. Bacteria commonly tend to do asexual reproduction, so they have one cell, and when it wants to reproduce, it just splits into two identical cells. Now, that is great for reproducing quickly. That, however, is not great for genetic diversity. It just results in multiple clones of the same organism, all genetically identical, unless something goes wrong and results in a mutation. And as we know, we like genetic diversity. It's kind of a good thing. Now, certain bacteria can also do something akin to sexual reproduction, which is the transfer of genes between two individuals by the use of a sex pilus. It is basically sexual reproduction for bacteria, essentially. But that doesn't happen in all bacteria, and a common way of creating genetic diversity is through horizontal gene transfer. So there are a few different mechanisms by which horizontal gene transfer can occur between species and individuals. So the first one is transformation, where after, for example, a bacteria dies, the cell lyses and bursts and releases all of its contents into the environment. So we have this dead cell and something needs to be done with it. So the cleanup crew comes, often other bacteria. They come and eat the dead cell, which includes the plasmids, or circular DNA bits, from the dead cell. Those plasmids contain genetic information, and if a cleanup cell comes to eat the plasmids, or clean up the bacterial debris, it eats some of those plasmids, and if it is permissible, it can integrate those plasmids into its own cell, into its own genome, and then begin expressing those new genes to get new functions. Another form of this is called transduction, which is kind of an indirect transfer of genes. So this happens is when the original bacteria cell gets infected by a bacteriophage virus. So the bacteriophage injects its own DNA into the host bacteria, does its virus things, and as it is replicating its genome, the viral genome, it picks up some of the bacterial genes. Then, when the virus finishes its life cycle in the bacteria, it bursts and releases new viruses, taking some of that bacterial DNA with it. Then, when the new bacteriophages infect a new bacteria, it can inject it. When it injects its own genome, it now injects both the viral genome and the picked up bacterial genome into the new bacteria. And if the new bacteria host replicates before cell lysis and bursting, it can then pass on those new bacterial genes to its offspring. There is also conjugation, just kind of as I mentioned before, the transfer of genetic material between bacteria by the use of a sex pilus, where plasmids from one go into the other and vice versa, and resulting in a newly recombined genome. Now, horizontal gene transfer is very interesting because it can happen between bacteria that are not very closely related at all. In order to transfer genes between individuals in eukaryotes, such as humans and other multicellular organisms, there often needs to be pretty closely related, but not in the case of bacteria. This can result in lots of interesting things happening, such as a previously harmless strain of bacteria becoming pathogenic, bacteria that were not previously resistant to a certain antibiotic gaining that antibiotic resistance, gaining evolutionary features such as resistance to desiccation or resistance to radiation, for example. And yeah, it can just recombine and make some fun and spicy new bacteria, not necessarily species, but at the very least strains. And so this also becomes an issue in medical research, such as, well, what happens if this bacteria gets this and then becomes resistant to all of our antibiotics? Uh-oh. That is a bit of an exaggeration, but a very real thing that has to be kept in mind when doing research on bacteria, especially in medical cases.